The Human Genome Project was a huge coordinated effort to identify the sequence of all the genes of the human genome. So there are still a lot of information that we're still sifting through. This was published back in like 2000, 2001, some of this information, and we're just now getting through a lot of this information. There were two competing interests. One was a public group that was trying to get this research done, and another was a private company that was trying to get this work done. And I think that a lot of this wouldn't have been done as quickly as it had by the public research had this company not kind of spurred them on. And there was a bit of a race, but they did the technique in different ways. They came up with slightly different results, but overall, both of them are very, very useful. We found the human genome contains 3.1 billion nucleotides, but the protein coding sequences only make up about 2% of the genome. It's a very small portion, right? What is all the rest of this DNA doing? We're still trying to figure that out. Is it regulatory? What else is it doing? The genome sequence is about 99.9% similar in individuals of all nationalities. However, there are single nucleotide polymorphisms, or SNPs, and copy number variations, or CNVs, which will account for genome diversity from person to person. The genome is dynamic. At least 50% of the genome is derived from transposable elements, things that can jump around in that genome, and other repetitive DNA sequences. The estimated number is about 20,000 protein coding genes. When they first started this project, they predicted much more, like 80,000 to 100,000 genes, just because humans are so complex. The fact that we only have about 20,000 protein coding genes is really just outstanding, especially if you consider how complex we are. Of course, there are different ways you can get different splicing and different regulation of genes, and that's going to lead to some of the differences that we see. The average size of human genes about 25 kilobases, and that includes, though, the regulatory regions, introns, and the exons. The mRNAs are closer to about 3,000 nucleotides long. Alternative splicing is much more important than we initially realized, and so this is what helps our human cells to produce a much, much larger variety of proteins. Some estimates are as many as 200,000 from only those 20,000 genes. So now we start to see, okay, well, this is where some of this complexity is coming from. About 50% or more of the human genes show a high degree of sequence similarity to genes in other organisms. So this is useful when we're doing things like comparative genomics, because we can use model organisms to study these genes, and they're going to have similar functions in both organisms. However, about 40% of the genes identified so far have no known molecular function. Maybe these genes are hugely important for things like metabolism. Maybe they're hugely important for things like cancer. I mean, we just don't know. The genes are not uniformly distributed on the chromosomes. We find gene-rich clusters that are separated by these gene-poor deserts that happen to account for about 20% of the genome. Chromosome 1 contains the largest number of genes, the Y chromosome the smallest number of genes. Human genes are larger, they contain more introns than genes in other organisms. Some of them are very, very large. Most of these are composed of introns. Most of it ends up being spliced out. Why is this? Who knows? Maybe you can be the scientist to discover some of these things. So we've talked about a few of these. Less than 2% of the genome codes for proteins, about 20,000 protein coding genes, although with alternative splicing that number might be tenfold that in terms of the number of different varieties of proteins that are produced. And that's leading to this alternative splicing. So what we do know is functions that have been determined previously, functions that are of genes that have been compared to known genes and predicted in protein sequences from other species, predictions based on annotation, analysis of protein, what their structure would look like, their functional domains, their motifs, have assigned a lot of these to different functional categories. So what you can see, again, about about a third or more of these genes, the molecular function is completely unknown. 41.7% of these. Many of them are involved with things like enzymes, signal transduction or passing signals through a cell, nucleic acid binding, things like transcription factors, DNA replication, and so on. Viral proteins. This is an interesting one. About 0.3% of our genome is actually composed of viral proteins. These are probably viruses, retroviruses, that have stably integrated into our genome, and they may provide some sort of a useful benefit to us. There's one viral protein in particular I know that's involved with adhering of the human embryo to the uterus of a woman who's pregnant. With this gene stably introduced into the human genome, these embryos are much more likely to stick to this uterus, and this is going to be thus much more successful for replication, and it's going to stick around in the human genome. Something like that is something that's going to be incorporated into the human genome. But again, a huge portion of these molecular function unknown. One of the difficulties with trying to figure out what are they doing? Like, there should be tons of people who are working on these. Is things like getting funding for studying a gene. Okay, we don't know what it does. Heck, we don't even know that it is a gene that's making a protein product for sure. How likely is a government to fund some research like that, especially with the way the funding environment is right now and the way the budget is right now? It's not going to happen. So identifying the function of many of these genes might take years. So the human genomic sequence is about 99.9% the same. Most of the genetic differences we see are going to result from the SNPs, or the single nucleotide polymorphisms. Many of these are associated with disease conditions, and there are several different projects going on trying to associate these different single base pair changes in the genome with different diseases and the likelihood of an individual to get these diseases. Copy number variants, just segments of DNA duplicated or deleted, and it's a similar idea with those.